All right, let's dive right into this explainer. Today, we're unpacking the absolute foundation of molecular biology, the incredible biological software that runs, well, every living thing. You can really think of the organisms around us, and obviously ourselves, as these highly complex biological machines. And the material we're looking at today, it's basically the master manual. We're going to explore the structure, the function, and the regulation of the absolute most basic biomolecules that make all of this possible.
Now, the source material kicks things off with this really inviting thought. Welcome to your journey on molecular biology. Because, this isn't just a dry list of scientific facts. It's a genuine journey into the molecular basis of our existence. It's asking us to shift our perspective entirely, to look at life through this incredibly microscopic, highly dynamic lens.
And the timeline for this journey? Just 15 weeks. In that time, the curriculum lays out a path from basically square one with biomolecules all the way up to the absolute cutting edge of genetic science. It's a really structured ascent, taking us from the raw basics to mastering the complex programming languages of life.
But why does this matter? Well, understanding cellular mechanisms and gene regulation is the absolute bedrock for diagnosing diseases and pushing modern medicine forward. The rationale is crystal clear here. You literally can't build the future of biotechnology, genetics, or biochemistry without this foundation. By getting a grip on the molecular roots of diseases, we open the door to creating actual real-world medical breakthroughs. It's huge.
So here's our roadmap for today. We'll start with the blueprint of life, move into copying the code, and then look at gene expression in action. After that, we'll cover glitches in DNA repair, and finally wrap the whole thing up with manipulating genetic code.
First up, Section 1. The blueprint of life, hardware and code.
Let's think about these foundational concepts as two distinct types of biological hardware running our software. The source highlights how crucial it is to understand the cellular organization of both prokaryotic cells, the simpler, usually single-celled organisms without a nucleus, and eukaryotic cells, which are the much more complex ones that make up plants, animals, and us. It's exactly on these two different hardware platforms that the entire history of life is played out.
And here we have the physical code itself, the nucleic acids and base pairs that actually store all this biological data. We're talking about the structure, the core properties, and the functional forms of nucleic acids. Getting your head around these nucleotides and how they form specific base pairings is honestly like learning the alphabet of life. It literally spells out the instructions for everything a cell needs to do across all the different forms of DNA and RNA. Now, to fully grasp how this code behaves, we need to look at how nucleic acids physically interact, specifically through denaturation and renaturation. Denaturation is basically the pulling apart of the DNA structure, usually because of heat or chemical changes, while renaturation is the incredible process of those strands perfectly zipping right back together. Understanding this physical flexibility, this resilience of DNA, is absolutely massive in molecular biology.
Moving right along to Section 2, Copying the Code, Replication Mechanics. So how does this software flawlessly duplicate itself? This dynamic mechanism of DNA replication can actually be broken down step by step, and it's all executed by specialized cellular machinery. First up, you need specific enzymatic roles, basically proteins acting as tiny biological motors to drive the whole thing. Then, it follows highly precise replication models in distinct stages. Plus, as the text notes, this entire process plays out differently depending on whether the DNA molecule is linear, like in our cells, or circular, like you'd find in a lot of bacteria. But here's the truly amazing part. The system has a built-in spell checker to ensure replication fidelity and avoid critical errors in the code. Seriously, think of it as a flawless biological autocorrect. As millions of base pairs are being copied, mistakes are bound to happen, right? But the cellular machinery actually double-checks its own work as it builds those new DNA strands, catching and correcting errors on the fly. It makes sure the new copy is as perfect as the original. How cool is that?
Which brings us to Section 3, Gene Expression in Action, Running the Program.
This is where the software actually executes. It happens in exact stages to turn that raw genetic code into targeted working proteins. First, you have transcription, where the DNA code gets transcribed into RNA. Next comes translation, where that new code is translated into actual proteins. But actually, it doesn't stop there. The source material makes a point to highlight post-transcriptional and post-translational modifications. These are essentially the final little tweaks and folding processes that make these proteins fully functional and ready to get to work in the cell.
Now, obviously, this biological program doesn't just run wild. It relies on highly precise regulatory systems to control exactly when and where genes are expressed. We see this in action through systems like operons in prokaryotic cells and the incredibly complex eukaryotic gene regulation happening in our own bodies. It's the reason a skin cell acts like a skin cell and a muscle cell acts like a muscle cell. It's all about turning specific parts of the genetic code on or off at the exact right millisecond.
All right, Section 4, Glitches and DNA Repair, when systems fail.
So what happens when the code breaks? Take a second to really think about that, because mutations in this code are the underlying basis of both evolutionary change and debilitating diseases. Even with that incredible replication fidelity we just talked about, things like environmental factors or random copying errors can absolutely cause the structural components of our DNA to break or mutate. Getting a handle on these types of mutations is central to the entire field.
To really understand this, we need to define a crucial process, mutagenesis. This is the origin and development of a mutation in DNA. By studying mutagenesis, scientists can figure out the exact origins and developmental pathways of these genetic alterations. It's basically the science of figuring out how and why our master blueprint sometimes goes wrong. But don't panic, because fortunately, our cells have robust, built-in repair mechanisms constantly working behind the scenes to patch these glitches. The curriculum makes a really strong point here. Life isn't just fragile, it is unbelievably resilient. These repair mechanisms are tirelessly scanning our DNA, identifying the errors introduced by mutagenesis, and fixing them to maintain overall genomic stability.
And that brings us to our final part. Section 5, Manipulating Genetic Code, Hacking Life.
This right here is the ultimate form of biohacking. It allows us to actively intervene in the very architecture of genetic life. Recombinant DNA technology takes all those principles we just went over, from the raw structure of the code to how it's naturally expressed, and uses that knowledge to literally combine and manipulate genetic material right in the lab. And the genetic toolkit we use for this is incredibly precise. We use restriction enzymes, which act like tiny molecular scissors to cut the DNA code exactly where we want. Then, vectors and gene transfer methods step in to deliver that brand new customized code straight into a cell. And finally, PCR techniques, polymerase chain reaction, are used to amplify that code so we actually have enough of it to study or use. It is a highly refined, heavily engineered process.
And here's the real payoff. Manipulating these systems leads to massive advances in immunology and biotechnology. It brings us right back to those vital medical applications we touched on at the start. By mastering recombinant technology and understanding things like immunoglobulins, the proteins acting as antibodies in our immune system, molecular biology leaps from theoretical science straight into real, life-saving medical applications.
So I'm going to leave you with this final, thrilling question to ponder. If we can now read, copy, and literally rewrite the fundamental software of life, what on earth will we program next? It's wild to think about. From understanding the basic hardware of cells to actively hacking the code with recombinant technology, the journey of molecular biology is accelerating at breakneck speed. Thank you so much for joining me for this explainer. Keep asking questions, keep thinking about the code, and keep exploring the fascinating blueprint of life.